In today's lecture, we're going to be talking about Taylorella and Bordetella, which, despite being part of the same bacterial family, are really different in terms of their host range. Both of these genera are gram-negative coccobacilli and are all considered biocontainment level 2. Although Taylorella, specifically Taylorella equigenitalis, is a notifiable and foreign animal disease in Canada. Taylorella are carboxyphilic facultative anaerobes, while Bordetella are obligate aerobes. They will not grow under anaerobic conditions. They're non-fermentative, and as a genus, they are highly contagious, so readily transmit between individuals. This is a gram stain of a pure culture of Bordetella species. These organisms can be highly pleomorphic, so they can be either rods, cocci, diplococci, or, as you can see here, take on an ovoid morphology. In this image, you can see the Leifson flagella stain on Bordetella bronchoseptica, one of the motile species of Bordetella, and I think you can see these flagella all around the organisms that allow them to move throughout their environment. This is a pure culture of Bordetella bronchoseptica on sheep blood agar, and I think you can appreciate the very small individual colonies seen on the third and maybe even the fourth streak of the plate. Taylorella is a host-associated genus. It's primarily associated with the genital tract of equines. Bordetella, on the other hand, is a respiratory pathogen, and we find it in many animal species, and it's found among both healthy and diseased animals. The organisms we're going to focus on today include pigs, dogs, rabbits, and people, but it has a very broad host range, so we can find it in others as well. While in general, organisms within these genera do not survive well in the environment, Bordetella bronchoseptica actually has an environmental reservoir in amoebas. Within the genus Taylorella, we have two species, Equigenitalis and Acinigenitalis, and the prefix acini refers to donkeys. Bordetella has 15 species that are recognized, and we can differentiate them based on a number of relatively simple biochemical tests. So Bordetella bronchoseptica is motile, while many other clinically relevant species are not. Bordetella avium will grow on Columbia agar, while Bordetella pertussis, the agent of whooping cough in people, does not. In Taylorella, the virulence factors are really a bit of a mystery. We just simply don't know. And I think we can probably attribute this to the fact that it really only causes disease in horses, and so the research in these organisms is just limited. Among Bordetella, the adenylate cyclase hemolysin is recognized. This inhibits phagocytosis and also the local immune response. A variety of Bordetella species produce dermonecrotic toxin. So we see this in Bordetella bronchoseptica and its impacts in the atrophic rhinitis syndrome. So it inhibits porcine osteoblasts and causes atrophy of the nasal turbinates. It's also required for Bordetella avium to be pathogenic. And among Bordetella generally, the amount of this toxin produced may be related to the virulence of that particular strain. Bordetella also produce fimbriae, which are involved in attachment and colonization. They produce tracheal cytotoxin, which damages ciliated tracheal epithelial cells. And they also have type 3 secretion systems for effector secretion. Bordetella pertussis, the human pathogen, also produces pertussis toxin. This is believed to be responsible for whooping cough, and pertussis toxin is composed of 5 two-part subunits. So 5 AB subunits together makes up the toxin. It ultimately leads to increased cyclic AMP levels within the cell, which has broad effects on homeostasis. It also has systemic effects. It's been shown to increase insulin levels in the body, and it inhibits the recruitment of white blood cells, and it affects chemokine production, so really interferes with the innate immune response. Taylorella equigenitalis is the cause of contagious equine metritis in horses. Bordetella avium we see as the cause of turkey coryza. Bordetella bronchoseptica we recognize in a variety of species. It's associated with atrophic rhinitis in pigs, canine infectious tracheobronchitis or kennel cough in dogs, a snuffles-like disease in rabbits, and really, it can cause respiratory infections in many animals. And then finally, briefly, we're going to talk about Bordetella pertussis, the cause of whooping cough in people. So first, contagious equine metritis, Taylorella equigenitalis. What we see in affected animals is vaginal discharge in mares 7 to 10 days after breeding, and then a return to estrus, so they fail to conceive. In stallions, we don't typically see clinical disease. These animals are generally silent carriers, and the organism lives in the smegma that accumulates around the urethral fossa. So it's transmitted venereally from a colonized or silently infected male animal to the female. And this venereal transmission can be either during natural mating or also artificial insemination. Taylorella acinigenitalis has rarely been associated with really very similar infections. In these images here, you can see pathological lesions associated with this organism in mares. So on the left, we have a mucoid vaginitis, and there's some sort of tenacious discharge that you can see dripping out here. And then on the right, we have uterine edema. So this causes metritis, it causes a return to estrus, and it's associated with this inflammatory process of the uterus. In Canada, this is a notifiable disease. So you need to contact the Canadian Food Inspection Agency. They conduct surveillance and will do trace outs to determine other animals who are potentially exposed or infected. Treatment relies on antimicrobials, as well as topical cleansing of the vulva and clitoris, including the clitoral fossa, which in females is an important site of carriage. We rely on careful selection of studs, screening of stallions to make sure they're not carriers, and also import controls to make sure that we don't bring it in from outside the country. If we have to import a horse from an endemic region, they must be tested and potentially also quarantined as well. Bordetella avium is the first Bordetella species we're going to talk about, and it is the agent of turkey coryza. This is an infection of typically the upper respiratory tract, and we see turkeys sneezing. A sneeze in a turkey is called snick, and so that's one of the most obvious clinical signs. There's also nasal discharge, which can be very mucoid and very tenacious. They'll have large strings of tenacious mucoid strands coming out of their nostrils, altered vocalization and mouth breathing. Because this organism is so highly infectious and so contagious, up to 100% of the flock can be affected. We typically see this affecting younger birds, so generally less than 4 weeks. And if you see a high proportion of the birds in a flock sneezing, this is highly suggestive of bordellosis or turkey coryza. Animals develop clinical signs following a 7 to 10 day incubation period. And generally speaking, although we can have a very high rate of morbidity, the mortality tends to be low. So Bordetella avium infections on their own are not generally lethal, but if we have other secondary bacterial infections, we can see higher mortality rates. Transmission is generally via direct contact. We can also see it surviving in the environment and spread through contaminated feed, water, or litter. While I said previously that Bordetella are not typically environmental organisms, this can survive for kind of 1 to 6 months. This is an organism that also causes infections in chickens, although they tend to be less severe than what's seen in turkeys. These infections are treated with antimicrobials, generally speaking, the tetracyclines, and it's controlled with both vaccination and biosecurity. On the right, you can see one of the classical lesions associated with Bordetella avium infections in turkey, and that is flattening of the tracheal rings, the tracheal cartilage. So on the left, we have a normal trachea, and on the right, we have a flattened trachea. This is coming from an infected bird. So classical lesion. In pigs, Bordetella bronchoseptica is associated with infections of the respiratory tract, particularly in young pigs. We can see pneumonia or also atrophic rhinitis. If we think back to our lecture on the Pastoralaceae, you'll remember that atrophic rhinitis is a polymicrobial infection caused by Pastorella multocida and Bordetella bronchoseptica. The way that we think these infections work is that Bordetella starts the infection. It causes damage to those mucosal tissues, which allows Pastorella multocida to proliferate. Toxins produced by Pastorella cause epithelial hypoplasia, atrophy of mucus glands, and osteolysis. And then the dermonecrotic toxin produced by Bordetella bronchoseptica inhibits osteoblasts. Ultimately, what we see is atrophy of the nasal turbinates and shrinking of the snout. Just to review, this is what that can look like. So our pig here on the left, you can see his snout is greatly shortened, some sort of extra wrinkles on top of the nose, indicating that those soft tissues are compressed with destruction of the bony tissues inside. And then on the right-hand side, we can see a pig with a deviated snout. So it's not always just sort of smushed and shortened. It can actually be deviated unilaterally. On cut section, this is what these lesions can look like. So starting with the image on the right, here we have a normal section of a pig snout. You can see these nice scrolling turbinates. And on the left, we have near complete obliteration of those turbinates. So very, very profound pathological changes. On the left image here, you can see bilateral atrophy of both the ventral and dorsal turbinates, and also deviation of the septum.